Okay, let's unpack this. Forget everything you thought you knew about cancer just being cells dividing too fast. This deep dive is about seeing cancer for what it truly is. An incredibly complex, almost perverse mastery of the fundamental rules of life. It's not just breaking the rules, it's rewriting them, turning the body's own systems against itself.
What's fascinating here, and maybe a bit unnerving, is that this isn't just some simple malfunction. It's a fundamental challenge to homeostasis, to that intricate balance of growth, life, and death that keeps us healthy.
Yeah.
And as we dive into this source material, really digging into cancer cell biology, especially pulling from Chapter 72, you'll see just how sophisticated and honestly how vulnerable this whole rogue process truly is.
Our mission today, and really your mission if you're tuning in for that elite understanding, is to cut through the immense complexity, the sheer density of this topic. We're taking this core source material, pulling out the absolute most important nuggets of knowledge, those surprising facts, those critical insights, so you can walk away with a mastery of cancer cell biology that feels like seeing the matrix behind the disease.
Yeah.
We want you to get it like the most informed professionals do, that essential shortcut without drowning in jargon.
And truly understanding cancer at this deep biological level. I mean, how these cells arise, how they manipulate their environment, how they spread, and crucially, why they defy treatment. It's absolutely fundamental.
Can't really grasp the why without it.
Exactly. It's the only way to fully appreciate the strategies and, let's be honest, the immense challenges of modern cancer therapies. It helps explain why our current treatments can achieve remarkable successes sometimes and frustrating failures in others. It's not random. There are biological reasons.
So think of this as getting inside the mind of the enemy, but, at the cellular and molecular scale. We're going to explore how cancer cells define themselves, where they come from, how they evolve, the critical cellular control systems they dismantle, how they build their own support systems, fuel their rapid growth, and finally, how science is trying to exploit these fundamental defects with cutting-edge treatments. Ready to jump in?
Let's do it. Let's start right at the beginning. How do we even define cancer? Because the source material makes it really clear that it's much, much more than just unregulated growth.
Right. Unregulated cell division is definitely the starting point. That's what people usually think of. But what takes a potentially harmless lump, a benign tumor, and makes it a malignant killer, the source points to 4 key characteristics that define a malignant neoplasm.
Precisely. So beyond that uncontrolled division, a malignant tumor has the ability to avoid cell death. Apoptosis. They just ignore it. They don't follow the body's instructions to die when they should.
They become immortal, in a sense.
In a cellular sense, yeah. Then there's tissue invasion, the ability to break through normal tissue boundaries and spread locally, busting out of their designated area.
Oh.
And finally, the most feared characteristic, metastasis, that capacity to travel to distant parts of the body and actually start new colonies, new tumors.
And that's the crucial distinction, isn't it?
Yeah.
A benign neoplasm, it has the unregulated growth. It can get quite large, maybe push on things.
Sure.
But it lacks that invasion and metastasis. It stays put. Malignancy is defined by those destructive spreading powers.
Absolutely. And the source also gives us a useful breakdown, just for context, of how cancers are typically named based on where they originate. Helps keep things straight.
Okay.
You have carcinomas, which arise from epithelial tissue sink linings, like your gut, your lungs, your skin. They're the most common type.
Right.
Then sarcomas from mesenchymal tissues. That's stuff like bone, muscle, fat, cartilage.
Okay.
And finally, the hematopoietic cancers, leukemias, lymphomas, and plasma cell dyscrasias, like multiple myeloma, these originate from blood-forming tissues. This classification tells us the initial home of the cancer.
Got it. Now, here's where it gets really interesting and maybe a bit counterintuitive. Despite the incredible diversity of cancers out there, the source emphasizes that they almost always begin from a single cell, one cell that acquires a genetic alteration. This is the concept of monoclonal origin.
Yeah, one cell goes rogue, basically. It gets that initial set of damaging changes and it becomes the ancestor, the founder of the entire tumor mass.
Okay, so the tumor lineage starts from just one bad seed.
Yeah.
But that doesn't mean all the cells in the resulting tumor are identical clones, right?
Yeah.
The source immediately brings in the counterpoint. tumor heterogeneity.
Absolutely not. That's a critical concept. As that single founder cell divides and the tumor grows, its descendants continue to pick up new genetic and epigenetic changes. It's not static.
Like evolution and fast forward within the tumor.
Exactly like that. Think of it like a branching tree. Different branches acquire different sets of mutations over time. So within the very same tumor, you'll find distinct populations of cells, each with its own molecular fingerprint, its own unique set of alterations.
And it's not just internal changes driving this diversity, is it?
Yeah.
The environment around the cells plays a huge role, too.
A massive role. The tumor microenvironment, as it's called, is incredibly complex. It's not just cancer cells floating in space. You've got surrounding normal cells, the extracellular matrix they sit in, infiltrating immune cells buzzing around, blood vessels, secreted factors.
It's a whole neighborhood.
A bustling and often dysfunctional neighborhood. And this environment isn't uniform throughout the tumor either. Cells in a low oxygen area behave differently than cells near a blood vessel. Cells interacting with one type of immune cell might evolve differently than those seeing another type. These extrinsic factors contribute significantly to that diverse mix of cells within a single tumor.
And the source is crystal clear on the direct, devastating consequence of this heterogeneity, especially when it comes to treatment.
This is why heterogeneity is such a major hurdle in fighting cancer effectively. When you apply a therapy, even a really good targeted one, it's highly likely that within that diverse population of tumor cells, a subset already possesses inherent resistance mechanisms. Maybe they have a mutation that bypasses the drug's target. Maybe they've just cranked up a different survival pathway. They're already prepared just by chance.
So the treatment might wipe out the vast majority of the sensitive cells.
But that resistant minority, yeah, they survive.
And then they just take over.
Exactly. They proliferate because they're cancer cells, and they repopulate the tumor mass. But now, the tumor is largely made up of cells resistant to the therapy you just used. It's a major, major driver of relapse and treatment failure. It really highlights why we need therapies that can overcome this inherent adaptability and diversity, or maybe hit multiple targets at once.
Yeah, that helps explain why just one type of drug often isn't enough, especially in advanced cancers. Okay, let's talk about how this single cell even starts down this path. The source contrasts some rare scenarios with the more typical process.
Right. In very rare cases, a single, really potent genetic alteration can be the primary driver of a cancer from very early on. These often involve genes we call oncogenes, sometimes first discovered because they were carried by animal tumor viruses.
Oh.
The source gives examples like the ABL gene alteration in chronic myeloid leukemia, CML, which creates that specific BCR ABL fusion protein, or certain BRAF mutations in melanoma. These are powerful drivers.
So kind of like a single Big Bang event setting things off.
More or less, yeah, relative to other cancers. But, and this is key, the source emphasizes this isn't the norm for most common human cancers. The typical scenario is a multi-step process.
Right.
It requires the accumulation of many genetic and epigenetic abnormalities over time, all within that single cell lineage. It's a stepwise acquisition of dangerous traits. Not one hit, but many hits.
And these acquired capabilities are things that normal adult cells simply don't do.
Yeah.
They're gaining new destructive functions.
That's right. They gain the ability to ignore controls on proliferation and differentiation. They stop acting like mature, specialized cells and kind of regress to a more primitive, constantly dividing state. They acquire the ability to invade surrounding tissues, to metastasize to distant sites, and critically, to induce angiogenesis that's building their own blood vessels to get fuel and oxygen.
But the body isn't just sitting there defenseless, right? It has built-in safeguards against all of this happening.
Exactly. Normal cells have extensive, really sophisticated safeguards. Think DNA repair systems to fix damage constantly, checkpoints to halt cell division if something's wrong, mechanisms to trigger cell death if the damage is irreparable, and systems to prevent cells from invading or growing out of place.
So cancer development is fundamentally about breaking through these multiple layers of defense. It's like a heist movie, bypassing one security system after another.
That's a great analogy, and the source describes this often as a progressive phenotype. You can sometimes see the changes happening over time in tissues, looking at precancerous lesions under a microscope.
Okay.
Yeah, many cancers follow a recognizable path. Maybe starting with simple hyperplasia, just too many cells, progressing to an adenoma, a benign tumor, then dysplasia, cells looking abnormal, disorganized, then carcinoma in situ, cancer cells, but they haven't broken through the basement membrane yet.
So containing.
Still contained, exactly. And finally, invasive cancer with the potential to metastasize. This whole progression often unfolds over many years, sometimes even decades.
That slow burn really highlights how many steps, how much accumulated damage is usually required for a full-blown cancer to emerge. So let's really get into the nitty-gritty of how cancer dismantles those critical cellular control systems. First up, the cell cycle, the process of how cells divide.
Okay, the cell cycle. It's a tightly regulated process fundamental to life with distinct phases. G1, that's growth and preparation for DNA copying. S, when DNA synthesis actually happens. G2, more prep, getting ready for division. And M, mitosis, the physical act of cell division. Cells can also exit into a resting state. G0.
And crucially, there are checkpoints at key transitions, like security guards making sure everything's okay before proceeding.
Precisely. You've got the G1S checkpoint before the cell commits to replicating its DNA, the G2M checkpoint before it divides, and the spindle checkpoint during mitosis itself, making sure chromosomes are divided correctly. These are critical quality control points.
And cancer cells somehow find ways to just blast right through these checkpoints.
They have to. A key mechanism is inactivating tumor suppressor genes. These genes normally act as brakes on the cell cycle. The source highlights two absolute central players here, real master regulators, RB and P53.
Let's focus on that G1 checkpoint first. Its job is basically to make sure the cell is ready, has the resources, and has fixed any DNA damage before it starts copying its entire genome in S phase. And RB, the retinoblastoma protein, is the central brake here.
Yes, exactly. RB normally works by binding to and inactivating transcription factors, specifically E2-FDP1. These factors are needed to turn on the genes required for S phase. So RB puts a lock on S phase entry.
So how does a normal cell get past RB to divide?
Good question. To pass this checkpoint normally, enzymes called cyclin-dependent kinases, or CDKs, have to phosphorylate RB. Adding that phosphate group changes RB's shape, making it release E2-FDP1. E2-FDP1 is now free to activate S-phase genes, and the cell cycle progresses.
And there are brakes on the brakes, negative regulators.
Right. Proteins like P21, P16, and P27 can inhibit these CDKs. They're like an extra layer of control, making sure the CDKs don't just phosphorylate RB recklessly.
And the source says something really striking here. Nearly every cancer has a defect somewhere in this G1 checkpoint pathway.
That's a critical insight, yeah. It tells you how fundamental this control point is, whether it's a mutated RB itself or an overactive CDK, or maybe the loss of a CDK inhibitor like P16 cancer must find a way to disable this G1 brake to achieve that relentless, uncontrolled proliferation.
Okay, so they find a way to bypass G1. What happens at the G2 checkpoint before the cell actually divides?
The G2 checkpoint is another quality control step. Its job is to ensure that DNA replication during S phase is complete and accurate, and that any DNA damage incurred during replication has been repaired before the cell enters mitosis, M phase.
And if damage is detected here?
If damage is detected at G2, or really at any point, the P53 pathway is typically activated. This brings us to that other master regulator.
P53, often called the guardian of the genome. This sounds incredibly important. How does this pathway, this crucial safety net, actually work?
It's a really elegant system. Normally, P53 protein levels are kept very low in the cell. It's constantly being bound by another protein called MDM2. And MDM2 acts like a tag, targeting P53 for degradation by the cell's protein disposal machinery.
So normally it's made and quickly destroyed.
Exactly, kept on a short leash. But when DNA damage occurs, say, from UV light or chemical mutagens, sensor proteins like ATM are activated. ATM then phosphorylates MDM2. This phosphorylation prevents MDM2 from binding to P53.
So the destruction tagger gets disabled.
Right. P53 is released from MDM2's grip. It's no longer being targeted for degradation, so it starts to accumulate in the nucleus.
And then this accumulated P53 swings into action. What does it do?
It has several options depending on the situation.
Yeah.
It can induce cell cycle arrest, often by activating the gene for P21, that CDK inhibitor we mentioned earlier, which helps halt the cell cycle, buying time for repairs.
Okay.
It can directly recruit DNA repair machinery to the site of damage, or, if the damage is too severe, too extensive to fix.
Self-destruct.
Exactly. P53 can initiate apoptosis-programmed cell death to eliminate the dangerously damaged cell before it can pass on mutations or become cancerous.
Wow. The source also mentions another way P53 gets activated, not just by DNA damage, but by dangerously high signals coming from oncogenes like MITE.
Yes, this is a fascinating backup system, sometimes called an oncogene checkpoint. If an oncogene like MITE becomes hyperactive, driving the cell towards uncontrolled proliferation, this excessive signaling can induce a protein called P14ARF.
Okay, and what does P14ARF do?
P14ARF competes with P53 for binding to MDM2. So it essentially distracts MDM2, preventing it from degrading P53. Again, P53 escapes destruction, accumulates, and can trigger either cell cycle arrest via P21 or apoptosis.
So it's a defense mechanism against cells becoming cancerous purely because an oncogene went haywire.
Precisely. It links abnormal proliferation signals directly back to the P53 safety net. The source also mentions senescence, that irreversible growth arrest, as another protective outcome that can be mediated by P53 in response to oncogenic stress.
So P53 is involved in blocking cells with DNA damage and cells with dangerous oncogene activity. It's like a double-layer safety system. And of course, cancer absolutely has to disable it.
It's almost essential for cancer progression. Mutations in the P53 gene itself, the gene is called TP53, are the single most common genetic alteration found across all human cancers, present in roughly half of all tumors.
Half? That's huge.
It is. Often, one copy of the gene is mutated and the other copy is lost entirely or silenced epigenetically. The source points out some interesting environmental links too, like a specific mutation pattern in P53 caused by aflatoxin exposure, which is common in hepatocellular carcinoma, or liver cancer.
And inheriting a faulty copy.
That causes Li-Fraumeni syndrome. Individuals born with one non-functional P53 copy have a drastically increased risk of developing multiple types of cancer, often at a very young age. It highlights how critical just one good copy of P53 is. Cancer cells can also disable P53 indirectly, maybe by overexpressing MDM2 or other negative regulators.
And this is also where certain viruses like high-risk HPV come into the picture, right? They target these pathways.
Absolutely. The high-risk human papillomaviruses, strongly linked to cervical cancer and others, produce viral oncogenes called E6 and E7. HPV E6 specifically binds to P53 and promotes its degradation.
It gets to the Guardian.
Exactly, and HPV E7 binds to and inactivates RB, that G1 break we talked about. So, by targeting these two critical tumor suppressors, RB and P53, these viruses effectively dismantle the body's primary defenses against uncontrolled epithelial cell growth. It's a key part of how they cause cancer.
Okay, wow. What about the final checkpoint in cell division, the spindle checkpoint during M phase, during mitosis itself?
Right, the spindle assembly checkpoint. During mitosis, the cell has to accurately segregate its duplicated chromosomes into two identical sets for the two daughter cells. This checkpoint monitors whether all chromosomes are correctly attached to the spindle fibers and properly aligned at the cell's equator before division proceeds.
Making sure each daughter cell gets exactly the right number of chromosomes.
Precisely. If there are errors like chromosomes aren't attached properly or the cell has the wrong number of chromosomes, a condition called aneuploidy, this checkpoint should ideally trigger cell death. This prevents the creation of genetically unstable daughter cells.
But cancer cells often are aneuploid. They frequently have wildly abnormal numbers of chromosomes.
That's right. Defects in the spindle checkpoint allow cells with the wrong chromosome numbers to survive and continue dividing. Aneuploidy is a hallmark of many, many cancers. In some tumors, the source notes this chromosomal instability, CIN, seems to be the predominant type of genetic chaos driving tumor evolution and heterogeneity. Bypassing this checkpoint basically facilitates this genetic instability.
So just to recap the damage, cancer breaks, the cell cycles breaks, like ARB. It disables the crucial DNA damage sensor and executioner P53, and it ignores the mechanisms that prevent errors in chromosome segregation during division.
It's a systematic dismantling of quality control. And that accumulation of errors also ties directly back to defects in DNA repair pathways themselves. The source lists a whole array of different pathways cells use to fix different kinds of DNA damage.
Yeah.
Mismatch repair for replication errors, pathways for fixing double-strand breaks like homologous recombination and non-homologous end joining, single-strand break repair, base excision repair for damaged bases, nucleotide excision repair for bulky lesions. It's complex.
And if these repair pathways themselves are defective in a cancer cell,
Then the mutations accumulate even faster. It leads to profound genetic instability, either through point mutations piling up, that's called microsatellite instability, or MIN, if mismatch repair is faulty, or through large-scale chromosomal abnormalities like aneuploidy, CIN. This instability fuels the heterogeneity we discussed earlier, giving the tumor more options to evolve and resist therapy.
The source gives examples like epigenetic silencing of mismatch repair genes, like MLH1 or MSH2, which is common in certain types of colon cancer and leads to MIN.
Right, and it's fascinating. The source mentions this tendency for tumors to often have either defects leading to MIN or defects leading to CIN as their primary mode of instability, but frequently not both being the main driver.
Suggests different routes to the same end goal of genetic chaos.
Seems like it. And from a therapeutic standpoint, this creates a vulnerability. Cancer cells with defective DNA repair are often more sensitive to therapies that cause DNA damage, like many traditional chemotherapies or radiation.
Because they can't effectively fix the damage you're inflicting on them.
Exactly. It's exploiting an inherent weakness created by their own defects. The source contrasts this exploiting a defect with the much greater challenge of therapeutically restoring a lost or broken tumor suppressor or repair function. That's much harder to do, although it mentions tools like CRISPR gene editing as potential future avenues for maybe attempting such restoration approaches someday.
Okay, let's move from controlling cell division to controlling cell death. Apoptosis. The source calls it a critical safeguard, which makes sense.
Absolutely. It's the body's elegant, programmed way of getting rid of cells that are damaged, aged, infected, or potentially dangerous, like cells that have accumulated mutations. For a cell to become cancerous and persist, it absolutely must acquire changes that allow it to subvert or bypass these programmed cell death pathways.
It needs to learn how to cheat death.
Pretty much. The source outlines two main pathways that trigger apoptosis, the extrinsic, or death receptor pathway, and the intrinsic, or mitochondrial pathway.
Okay, tell us about the extrinsic pathway. Triggered from outside.
Yes. Certain immune cells, for example, can trigger it. Some cells express death receptors on their surface, proteins like FAS or DR4-DR5. When specific signaling molecules, called ligands, like FASL or TRAIL, bind to these receptors, it initiates a cascade inside the cell.
What happens then?
Adaptor proteins are recruited, and they activate an initiator caspase called procaspase 8. Activated caspase 8 then activates downstream effector caspases. These are the real executioners, like caspase 3 and caspase 7. They go on to dismantle the cell in an orderly fashion, chopping up proteins and DNA.
Okay, so that's death triggered by an external kill signal. What about the intrinsic pathway, triggered from within?
Yes, this pathway responds to internal cellular stresses, things like severe DNA damage, loss of adhesion to the surrounding tissue, a specific type of apoptosis called anoikis, excessive signaling from oncogenes, or lack of essential growth factors. These stresses send signals to the mitochondria.
The cell's power plants, they're involved in cell death too.
They are central regulators of this intrinsic pathway. When stressed, the mitochondria release key proteins that are normally kept inside them out into the cytoplasm. The main ones are cytochrome C and another protein called SMAC, or Diablo.
And what do these release proteins do in the cytoplasm?
Cytochrome C binds to other proteins, APAF1 and procaspase 9, to form a complex called the apoptosome. This complex activates caspase 9, another initiator caspase, which then activates the same effector caspase 3, NIF7, as the extrinsic pathway.
Okay, what about SMT?
SMAC plays a cool role. There are proteins in the cell called IAPs, inhibitors of apoptosis proteins, that normally act as brakes, inhibiting caspase activity. SMAC binds to and neutralizes these IAPs, essentially removing the brakes on the death process.
So the mitochondria are holding the trigger, and internal stresses pull it by releasing cytochrome C to activate caspases, and SMAC to remove the inhibitors. The source says the balance of proteins from the BCL2 family is what determines whether the mitochondria release these factors.
This is the crucial regulatory point for the intrinsic pathway. The BCL2 family is a whole group of proteins, some of which are anti-apoptotic, they promote survival, and some are pro-apoptotic, they promote death.
Like a tug of war.
Exactly. The anti-apoptotic members, like BCL2 itself, BCLXL, and MECCL1, work primarily by binding to and sequestering the pro-apoptotic members. The pro-apoptotic side includes BH3-only proteins like BAD, BIM, BID, PUMA, and NOXA, which act as sensors of cellular stress. When activated by stress, These BH3-only proteins can either neutralize the anti-apoptotic BCL2 members or directly activate another group of pro-apoptotic proteins called BAX and BAC.
And BAX and BAC are the ones that actually punch holes in the mitochondria.
Essentially, yes. Activated BAX and BAC oligomerize and form pores in the outer mitochondrial membrane, which allows cytochrome C and SMAC to leak out into the cytoplasm, triggering the caspase cascade.
So the cell's fate, live or die via the intrinsic pathway, really hinges on the balance, the ratio, between these pro- and anti-apoptotic BCL2 family members. Cancer cells must tip that balance towards survival.
They absolutely do. A common strategy is to overexpress anti-apoptotic members like BCL2 or BCLXL, or they might inactivate pro-apoptotic ones, sometimes indirectly by disabling p53, which normally activates some BH3-only proteins like PUMA and NOXA in response to DNA damage. Overexpression of BCL2, for example, is very common in many lymphomas, leukemias, and also seen in prostate, breast, lung cancer, melanoma.
And this dependency creates a vulnerability, right? An opportunity for treatment. The source mentions targeting these pathways.
Yes, because many cancers become reliant, even addicted to these overexpressed anti-apoptotic proteins for their survival. This led to the development of drugs called BH3 mimetics. These drugs mimic the action of the pro-apoptotic BH3-only proteins.
How do they work?
They bind specifically to anti-apoptotic proteins like BCL2, essentially prying the pro-apoptotic proteins free, or preventing BCL2 from sequestering them. This tips the balance back towards death. The source mentions venetoclax, a specific BCL2 inhibitor, which is approved for certain types of leukemia like CLL with a 17p deletion, which affects p53, and shows activity in AML. It's a prime example of rationally targeting an apoptosis defect.
Are there other ways to target apoptosis?
Well, trying to trigger the extrinsic pathway using agonists for the death receptors, like trail agonists, has been explored, but the source notes clinical activity has been somewhat limited so far, maybe due to resistance mechanisms or delivery issues.
And you mentioned other general survival pathways are also frequently hijacked, like PI3K actin and SP. These also help cancer cells avoid apoptosis.
Definitely. These major signaling pathways, when hyperactive in cancer, send strong pro-survival signals. The PI3-ACT pathway, for instance, can phosphorylate and inactivate pro-apoptotic proteins like BAD, or activate transcription factors that promote the expression of anti-apoptotic proteins. Inhibitors targeting PI3K or ACT are in development, but as the source notes, because these pathways are also important in normal cells, toxicity can be a significant challenge.
Let's look closer at NFP. The source gives a detailed description of that pathway, which is apparently important for understanding how a certain class of drugs works, especially in myeloma.
Right. NFK is a key transcription factor involved in inflammation, immunity, and cell survival. It's usually a complex of two proteins, typically P65 and P50. In its inactive state, it's held sequestered in the cytoplasm because it's bound to an inhibitor protein called IB.
Okay, so IKEY keeps it locked up. How does it get released?
Signals that activate NFP, like inflammatory cytokines or growth factor signals, trigger the activation of an enzyme complex called IKK, I-pinase. IKK does exactly what its name suggests. It phosphorylates IP.
And phosphorylated IB is then targeted for destruction.
Yes. It gets tagged with ubiquitin and degraded by the proteasome, the cell's protein recycling machinery. Once IB is degraded and gone, NFD is free. It can then translocate, move into the nucleus, and turn on the expression of a whole host of genes, including many that promote cell survival and proliferation and suppress apoptosis.
Ah, okay. And proteasome inhibitors, like those used effectively in multiple myeloma, exploit this dependence.
This is a really elegant example of targeting a specific cellular process. Proteasome inhibitors, the source mentions bortezomib, carfilzomib, and ixazomib, block the activity of the proteasome.
So the cell's garbage disposal gets jammed.
Exactly. And one critical consequence of jamming the proteasome is that phosphorylated IB doesn't get degraded. It stays bound to NFK, trapping the NFK complex in the cytoplasm.
So you lock NFK out of the nucleus, preventing it from turning on those survival genes.
Precisely. Without that crucial NFP activity driving survival, myeloma cells, which are often highly dependent on it, become much more susceptible to undergoing apoptosis. It's a key mechanism of action for these drugs. The source also mentions that inhibitors targeting IKK itself are in development, aiming to block the pathway further upstream.
Very clever. So by blocking the proteasome, you indirectly shut down a key survival pathway.
It's a great example. And the source also points out just briefly that epigenetic silencing of key apoptosis genes like APAF1, part of the apoptosome, or caspase-8, the initiator for the extrinsic pathway, is another trick cancer uses to evade death. This adds yet another layer of complexity and potential therapeutic targets.
It's truly astonishing, the multi-pronged approach cancer takes to ensure its survival and just completely disregard the body's normal controls for growth and death.
And this disregard extends beyond just the cell's internal workings. Cancer cells also ignore the signals that tell them to stay in their designated place, to respect tissue boundaries, and to stop growing when appropriate. The source describes cancer almost like an organ that has lost its specialized function and completely ignores the rules of tissue homeostasis.
Normal tissues have incredible regenerative capacity, right? Your liver can regrow after partial removal, your skin constantly renews itself, but they know when to stop. They stop when the job is done. Cancer just keeps going.
They've lost the ability to respond to those complex feedback loops that regulate tissue size, shape, and function in a normal organ. While we understand some aspects, the full picture of exactly what these stop growing now signals are and how cancer manages to bypass all of them is still an active and really important area of research.
And tumor growth itself isn't necessarily a simple, ever-accelerating process either. The source mentioned something called Gompertzian kinetics. What's that about?
Yeah, Gompertzian kinetics refers to the observation, often seen in experimental models and clinically, that as a tumor grows larger, its overall growth rate actually tends to slow down. This doesn't mean it stops growing, but the percentage of cells within the tumor that are actively dividing at any given time, what's called the growth fraction, decreases as the tumor mass increases.
Why does that happen? Is it just running out of space or fuel?
It's likely due to limitations imposed by the tumor microenvironment as the tumor gets bigger. The source points to factors like developing areas that are hypoxemic, low oxygen because the blood supply can't keep up, or having inadequate nutrients in the tumor core. Also, some cells might accumulate so much genetic damage that they can survive but can no longer proliferate. Effectively, they contribute to the mass, but not the exponential growth.
But then, here's the kicker. If you shrink the tumor with treatment, its growth rate can actually accelerate again.
That's a crucial and often frustrating clinical observation, the source highlights. Unlike normal tissues that stop proliferating once a repair or regeneration goal is met, the tumor seems to sense the reduced cell numbers, or maybe the improved conditions, like more oxygen and nutrients reaching the remaining cells after debulking. Surviving cells, including potentially those that were previously quiescent or slow-growing, can re-enter the cell cycle and start dividing faster again.
But unlike normal tissue, they don't stop when they reach the original size or density.
No, they just keep expanding relentlessly. They've lost that feedback control.
This dynamic interaction between tumor size, the microenvironment, nutrient availability, and growth rate is really important for thinking about treatment strategies, dosing schedules, and understanding patterns of recurrence.
Absolutely. And it underscores again how critical that tumor microenvironment is. It's not just passive scaffolding, it's an active participant influencing tumor behavior, growth rate, and even response to therapy. Massively important. And it's not just the physical constraints. Normal tissues also have ways to eliminate cells during development or homeostasis, like mechanical processes or cell extrusion, getting rid of cells that aren't needed. Cancer cells, due to their defects in cell cycle control, apoptosis, and signaling, manage to subvert these elimination mechanisms too.
Okay, let's delve deeper into that microenvironment itself. It's clearly more than just cancer cells. What else is in there?
Far from it. The source details a really complex ecosystem. Beyond the malignant cells themselves, you have a whole host of other cell types. Immune cells like lymphocytes, macrophages, myeloid-derived suppressor cells. You have fibroblasts, which make up the connective tissue, adipocytes or fat cells, endothelial cells forming blood vessels, lymphatic vessels, the extracellular matrix, ECM itself, which provides structure, and a whole soup of secreted factors like growth factors, hormones, cytokines, even reactive oxygen and nitrogen species. There are mechanical forces too.
It sounds like a whole bustling, interactive community.
It is, but a highly dysfunctional one, co-opted by the cancer. And importantly, this environment isn't static. It's dynamic, constantly evolving and interacting with the tumor cells. The source emphasizes that this microenvironment plays a significant role in helping cancer cells resist therapies through various mechanisms, providing survival signals, physically blocking drug access, suppressing immune responses. We're still uncovering all the ways it contributes.
Wow. Let's shift back for a moment to another fundamental cellular stop signal that cancer has to bypass: cellular senescence. What exactly is that?
Senescence is basically an irreversible state of growth arrest. The cell stops dividing permanently, but it doesn't die immediately. It remains metabolically active, often secreting various factors. It was first really characterized in normal cells grown in culture in the lab.
Normal cells don't just divide forever in a petri dish.
No, they don't. Normal human fibroblasts, for example, typically undergo a limited number of divisions, the source mentions around 30 to 50, before they enter this state called replicative senescence. They just stop dividing. This growth arrest is often triggered by an increase in those CDK inhibitors we talked about, like P21.
And the primary trigger for this replicative senescence in normal cells, the built-in counting mechanism, is telomere shortening.
This is a brilliant piece of biological engineering. DNA polymerase, the enzyme that copies DNA, has a quirk. It can't fully replicate the very ends of linear chromosomes, the telomeres. Telomeres are like protective caps on chromosome ends, made of repeating DNA sequences, TTAG, in humans, bound by specialized proteins, often forming a loop structure.
So, with each round of cell division,
a little bit of the telomere sequence is lost from the end. It gets progressively shorter. They act like a cellular odometer, counting division.
Clever. And what happens when they get too short?
When telomeres become critically short, the unprotected chromosome ends are recognized by the cell's DNA damage surveillance system, particularly the p53 pathway. It looks like a persistent DNA break. This triggers a stable growth arrest senescence, or sometimes apoptosis, often because these uncapped chromosomes can start fusing together, leading to genomic chaos if the cell tried to divide again.
Oh, yeah. So telomere shortening acts as a natural barrier, preventing normal cells from dividing excessively and potentially accumulating dangerous mutations. But cancer cells aiming for immortality must bypass this barrier.
It's absolutely critical for malignant transformation. The vast majority of cancer cells bypass this replicative limit by reactivating an enzyme called telomerase.
Telomerase, that's the enzyme that adds the repeating sequences back onto the telomeres.
Exactly. Most normal somatic cells in adults have very low or undetectable telomerase activity, so their telomeres naturally shorten with age and division. But cancer cells manage to turn telomerase expression back on, often at high levels. This allows them to maintain their telomere length or even lengthen them, effectively erasing the odometer and allowing them to divide indefinitely, bypassing replicative senescence.
The source is over 90% of human cancers express high levels of telomerase. That's a remarkably consistent feature.
It really is, which makes telomerase a very attractive, almost universal cancer-specific target for therapy. Efforts have been ongoing for years to develop telomerase inhibitors, or perhaps use components of telomerase, like its protein subunit H, as antigens for cancer vaccines.
Is there a downside to targeting telomerase?
The potential catch, noted in the source, is that some normal cells do need telomerase activity. Specifically, our stem cells, like those in the bone marrow, the gut lining, skin epidermis, and germ cells, rely on telomerase to maintain their own telomeres because they need to divide throughout life to replenish tissues. So, inhibiting telomerase systemically could potentially cause toxicity in these normal, renewing tissues.
That's a tricky balance. The source also mentions that other stresses, not just reaching the end of the telomere fuse, can also induce senescence.
Yes, it's not just about counting divisions. Things like DNA damage from radiation or chemotherapy, exposure to reactive oxygen species, or even the strong aberrant signaling from certain oncogenes can trigger a DNA damage response that leads to senescence, often involving p53 and p21 activation, similar to what happens with critically short telomeres.
It's acknowledged, though, that the exact relevance of senescence to what happens in human cancer in vivo inside the body is still being fully worked out.
There are complexities and perhaps paradoxes. The source mentions dyskeratosis congenita, a rare inherited disease caused by defects in telomere maintenance genes. You might expect severe problems in rapidly renewing tissues like the gut in these patients, but surprisingly, the GI effects aren't always the most prominent feature. So there's still much to unravel about telomere biology and its precise role in human aging, illness, and cancer initiation and progression in vivo.
Okay, let's shift gears now to how cancer cells communicate, how they receive signals from their environment, and how those signals dictate their behavior. We're talking about signal transduction pathways.
Right. Cells are constantly bombarded with signals from their surroundings, signals from adjacent cells, from the stromal cells in the microenvironment, from hormones circulating in the blood, sometimes even signals they produce themselves, which is called autocrine signaling.
And these signals need to be translated into actions inside the cell, right? Like deciding whether to divide or move or survive or differentiate.
Yeah, exactly. That's the job of signal transduction pathways. They act like intracellular communication networks. Typically, it involves a cascade or relay race where one protein activates the next, which activates the next, often through a process called phosphorylation.
Adding phosphate groups as on or off switches.
Precisely. Kinases are the enzymes that add phosphate groups, often acting as on switches for the next protein in the chain, and phosphases are enzymes that remove them, acting as off switches. These cascades can amplify the original signal significantly. The source highlights two major classes of kinases heavily involved in cancer: tyrosine kinases and serothionein kinases. Some kinases, like MEK, can even do both.
And the source specifically calls out two crucially important pathways that are frequently messed up in cancer: the ARAS-RAF-IC-ERK pathway, often called the MPK pathway, and the phosphatidylinositol 3 kinase, or PI3K pathway. And it notes these pathways are complex and interconnected.
These two pathways are central regulators of fundamental cellular processes, things like proliferation, growth, survival, migration, angiogenesis. They're absolutely key. Normally, the activity of the kinases within these pathways is very tightly controlled, usually short-lived, only active when the initial signal is present.
But in cancer, they get stuck in the off position, right?
Yeah.
Leading to constant signaling.
Exactly. This aberrant persistent activation is a hallmark of cancer. It can happen through various mechanisms: mutations in the cell surface receptors, often receptor tyrosine kinase or RTKs, that initiate the signal, or mutations in components downstream in the pathway itself, like RAS or BRIF or PI3K. It can also happen through gene amplification, where the cell makes way too many copies of a receptor or signaling protein, or through chromosomal translocations that create abnormally active fusion proteins.
And because these pathways are so central to driving cancer growth and survival, targeting them with drugs has become a major therapeutic strategy. This brings us squarely into the realm of targeted therapies.
Yeah, this field has absolutely revolutionized cancer treatment over the last couple of decades. The core idea is, if you can identify the specific molecular alteration that's driving a particular tumor, that specific broken protein stuck in the on-APO position, maybe you can design a drug, often a small molecule inhibitor or a specific antibody, that precisely blocks the activity of that aberrant protein.
Hitting the cancer where it hurts, specific.
Exactly. The source provides a whole slew of examples illustrating this principle. You have EGFR inhibitors like osimertinib, erlotinib, afatinib, used for lung cancers that have specific activating mutations in the epidermal growth factor receptor.
Okay.
Then there are inhibitors for other kinases, often rearranged or mutated in lung cancer, thyroid cancer, and others: ALK, ROS1, NTRK, MET, RET inhibitors. Drugs like crizotinib, alectinib, entrectinib, selpercatinib fall into this category.
And BRF inhibitors for melanoma.
Right. BRAF inhibitors like vemurafenib and dabrafenib have had a huge impact in melanoma patients whose tumors carry the specific V600E BRAF mutation. BRAF is a key kinase in that RAS-RAF-MEK-ERK pathway. Interestingly, these are often now used in combination with MECAR inhibitors like trametinib or cobimetinib, which target the next step down in the same pathway. Hitting 2 points helps overcome resistance.
Makes sense. What else?
JT inhibitors like ruxolitinib, used for myeloproliferative syndromes driven by activating mutations in the JAK2 kinase. And then there's the really groundbreaking drug, imatinib.
The CML drug.
Yes, Gleevec. It was a true paradigm shift. It targets the specific BCR-ABL fusion protein that drives chronic myeloid leukemia. But it also hits other targets like the SIGKIT receptor in gastrointestinal stromal tumors, GIST, and PDGFR in other cancers. It proved the concept that targeting the specific molecular driver could lead to dramatic responses. The source also mentions the development of second and third generation inhibitors like dasatinib, nilotinib, bosutinib, and ponatinib, designed specifically to overcome resistance mutations that emerged against imatinib, like the notorious T315I mutation in BCR-ABL. Ponatinib works against that one.
So constantly adapting the drugs to stay ahead of the cancer's adaptation.
It's an ongoing battle. There are also broader spectrum multi-kinase inhibitors, drugs like sorafenib, sunitinib, regorafenib, lenvatinib, cabozantinib, which hit several relevant RTKs involved in tumor growth and angiogenesis. These are used across a range of cancers like kidney, liver, thyroid, and colorectal cancer.
And the PI3K pathway hasn't been ignored either.
No, definitely not. It's a major focus. The source lists inhibitors targeting specific isoforms of PI3K itself, like idelalisib targeting the delta isoform, which is important in lymphoid malignancies, or alpelisib targeting the alpha isoform, PIK3CA, when it's mutated, particularly in breast cancer. Then there are MTOR inhibitors, everolimus, temsirolimus, which hit a key downstream node in the PI3K pathway. These are approved for kidney cancer, certain neuroendocrine tumors, and breast cancer.
And even inhibitors targeting the cell cycle machinery directly, like the CDK46 inhibitors.
Yes, drugs like palbociclib, ribociclib, and abemaciclib. These specifically inhibit cyclin-dependent kinases 4 and 6, which, as we discussed, are responsible for phosphorylating RB and releasing that brake on the G1 checkpoint. These have become standard of care in certain types of breast cancer, often combined with hormonal therapy.
What all these examples really hammer home is the incredible shift towards personalized or precision therapy in oncology.
Absolutely. The source highlights this clearly. Assessing a patient's tumor for specific molecular alterations, looking for those mutations, gene amplifications, translocations, or patterns of protein overexpression, is now essential, standard practice, to guide treatment decisions, especially in the metastatic setting for many cancers like lung, breast, melanoma, colorectal, and others. We match the drug to the specific driver in that patient's tumor.
And the rise of liquid biopsies, using circulating tumor DNA, ctDNA found in the blood, is making this personalized approach even more accessible and dynamic.
It's a huge advance. It's incredibly valuable.
Sure.
As cancer cells die, they release fragments of their DNA into the bloodstream. Detecting and sequencing this ctDNA allows us to find those targetable mutations without always needing an invasive tissue biopsy.
And potentially gives you a broader picture of the tumor's heterogeneity than a single biopsy might, right?
Exactly. A single needle biopsy might just sample one subclone within the tumor, whereas ctDNA might represent a mix from different tumor sites. It's being used now to monitor how well a treatment is working, to detect the emergence of resistance mutations much earlier than before, sometimes predicting relapse before it's visible on scans, and the hope is, as the technology gets even more sensitive and specific, it could be used for early cancer detection and screening programs.
But there's always a but with cancer. Despite these incredible targeted therapies, the source is quite realistic. They alone are rarely curative. CML treated with imatinib is mentioned as a notable exception, with high long-term survival rates, but that's still relatively rare across all cancers. Resistance almost always develops eventually.
This remains the persistent central challenge. Cancer cells are masters of adaptation. Resistance can occur through a multitude of mechanisms. A common one is a new mutation popping up in the target kinase itself, where the drug binds, so the drug can't attach effectively anymore. The T359 mutation in BCR-ABL blocking imatinib is the classic example.
So the lock changes shape, and the key doesn't fit.
Right. Or the cancer cell might just figure out how to activate an entirely different pathway to achieve the same result, survival and proliferation, effectively bypassing the roadblock you've put up.
So you hit pathway A, and the cancer just cleverly reroutes all the essential traffic through pathway B.
Precisely. Understanding these diverse resistance mechanisms is a major focus of research because figuring out how resistance happens allows us to develop rational strategies to overcome it, maybe using combinations of drugs to block the primary pathway and the escape route, or developing next-generation drugs that can hit the mutated target. The source also notes that sometimes the effectiveness of targeted drugs is limited simply by off-target toxicity side effects on normal cells that prevent using a high enough dose.
Okay. The source also mentions antibody-drug conjugates, or ADCs, as another sophisticated targeted approach, using antibodies basically as smart delivery systems.
This is a really brilliant concept. You take a monoclonal antibody that's designed to specifically recognize and bind to a protein that's highly expressed on the surface of cancer cells, but maybe less so on normal cells. Then you chemically link a highly potent cytotoxic drug, a chemotherapy agent, to that antibody via a stable linker.
So the antibody acts like a guided missile.
Exactly. The antibody circulates, finds the cancer cells expressing the target protein, binds to them, and gets internalized. Once inside the cell, the linker is often cleaved, releasing the potent chemotherapy payload directly into the cancer cell, killing it from within. This aims to maximize the drug concentration where it's needed and minimize exposure and toxicity to healthy tissues.
And there are examples of these in use.
Yes. The source lists several successful ADCs. Brentuximab vedotin targeting CD30 in certain lymphomas, adotrastuzumab emtansine, TDM1, and more recently famtrastuzumab deruxtecan targeting HER2 in breast and gastric cancers, gemtuzumab ozogamicin targeting CD33 in AML, inotuzumab ozogamicin for CD22 in all, polatuzumab vedotin targeting CD79B in lymphoma. It's a rapidly growing field.
And across all these strategies, the source emphasizes the increasing importance and use of combinations. It's often not just one drug anymore.
Right. Single agents, whether chemo or targeted therapy, are often insufficient to achieve durable control, especially in advanced disease, largely due to that heterogeneity and resistance problem. Combining targeted agents with each other, or with traditional chemotherapy, or now increasingly with immunotherapy, can potentially enhance effectiveness, overcome or delay resistance, or hit the cancer from multiple angles simultaneously.
Are there examples where combinations have clearly improved things?
Definitely. The source gives examples like adding targeted antibodies like trastuzumab against HR2 and often pertuzumab, another HR2 antibody, to chemotherapy for patients with HR2 positive breast and stomach cancers. These combinations have significantly improved survival rates, including in the adjuvant setting, meaning given after surgery to prevent recurrence. Another major example is adding rituximab, an antibody targeting the CD20 protein on B cells, to standard chemotherapy regimens for diffuse large B-cell lymphoma, DLBCL, which essentially revolutionized the cure rates for that disease.
That's really powerful.
Mm.
This leads us nicely into two incredibly important concepts for thinking about how we exploit cancer's fundamental weaknesses, concepts mentioned prominently in the source: oncogene addiction and synthetic lethality. These sound like ways to really target the core vulnerabilities.
They are. Oncogene addiction is one of the most insightful concepts, really explaining why targeting a single specific mutated protein with a drug can sometimes have such dramatic, profound effects on a cancer cell.
Okay, unpack that a bit more. What does addiction mean here?
It means that when an activating mutation occurs in a key oncogene, like BRAF in melanoma or EGFR in lung cancer, that single altered pathway can become the dominant, absolutely essential signal driving the cancer cell's survival and proliferation. The cell becomes profoundly dependent, addicted to the continuous high-level activity streaming from that specific pathway. It might even downregulate or become less reliant on other parallel pathways.
So it's like the cancer cell streamlines its operations and puts all its essential survival functions onto this one superactive pathway. It puts all its eggs in one basket.
That's a perfect way to think about it, and that extreme dependency creates a critical vulnerability. If you can then come in with a drug that specifically inhibits that one addictive pathway that the cell now relies on so heavily,
The whole system collapses.
Exactly. The cancer cell is much more susceptible to dying compared to normal cells, which aren't addicted, or even compared to cancer cells that might have the same mutation but haven't developed that same level of profound dependency. The source uses the example of BRAF mutated melanoma cells being strikingly sensitive to inhibitors of MEK, which is downstream of BRAF, as a prime clinical illustration of this oncogene addiction principle in action.
So that dependency, that addiction, is actually an Achilles heel created by the cancer's own primary driving mutation.
Yep.
Okay, what about synthetic lethality? It also sounds like exploiting dependencies, but maybe in a different way.
Synthetic lethality is a fascinating genetic concept, first described decades ago. It refers to a situation where a defect or loss of function in either of two genes individually is tolerated by the cell, that the cell survives just fine. But if you have simultaneous loss of function in both of those specific genes, the combination is lethal to the cell.
Okay, so losing A is fine, losing B is fine, but losing A and B together is deadly. Like having two redundant systems. If one fails, the backup kicks in, but if both fail simultaneously, the whole system crashes.
Precisely. And the therapeutic application in cancer is incredibly clever. Cancer cells, as we've discussed, very often have defects in tumor suppressor genes whose normal function is lost. These defects are hard to fix therapeutically. You can't easily just add back a working gene.
Right.
So the idea of synthetic lethality is to find another gene, let's call it gene B, that has a synthetic lethal relationship with the specific tumor suppressor defect already present in the cancer cell. Let's call that defect loss of gene A. If the cancer cell has already lost function of tumor suppressor A, it often becomes abnormally dependent on the backup pathway mediated by gene B for its survival.
So the loss of A makes it addicted to B.
Exactly. So now, if you use a drug to inhibit the function of gene B, it's synthetically lethal specifically to the cancer cells, which lack both A and B function. Normal cells, which still have a working copy of gene A, can tolerate the inhibition of gene B because they still have the functional pathway A as a backup.
That is incredibly elegant. You're exploiting the pre-existing tumor suppressor loss. And the source gives the key clinical example that really brought this concept into the mainstream: the relationship between BRCA mutations and PRP inhibitors.
This is the canonical textbook example now. BRCA1 and BRCA2 are critical tumor suppressor genes involved in a major DNA repair pathway called homologous recombination, HR, which is particularly important for fixing DNA double-strand breaks accurately. In cancer cells that have mutations inactivating BRCA1 or BRCA2, this HR pathway is defective.
Okay, so they can't efficiently repair double-strand breaks using that main pathway.
Right. As a consequence, these BRCA mutant cancer cells become highly reliant on other DNA repair pathways to survive, particularly pathways involved in repairing single-strand DNA breaks. One key enzyme involved in single-strand break repair is PRP, poly-ADP ribose polymerase.
So the BRCA mutant cancer cell, which has broken HR repair, is now abnormally dependent on PRP activity for its survival.
Precisely. Now, what happens when you treat these cells with a PRP inhibitor drug? The drug blocks PRP's ability to help repair single-strand breaks. These unrepaired single-strand breaks often get converted into double-strand breaks when the cell tries to replicate its DNA.
And the BRCA mutant cell can't fix those double-strand breaks effectively because its HR pathway is broken.
Exactly. The cell gets overwhelmed with unrepaired double-strand breaks, leading to catastrophic chromosomal damage, genomic instability, and ultimately, cell death.
Whereas normal cells, which have functional BRCA and working HR repair, can tolerate the PRP inhibition because they can still effectively repair the resulting double-strand breaks using the HR pathway.
Precisely. That's the synthetic lethal interaction. This is why PRP inhibitors, the source mentions olaparib, rucaparib, niraparib, and talazoparib, have shown significant efficacy and gained approval specifically for treating cancers with BRCA mutations, such as certain ovarian, breast, prostate, and pancreatic cancers. It's a beautiful example of exploiting a tumor-specific genetic defect.
Wow. So instead of trying to fix the broken tumor suppressor gene, BRCA, you kill the cell by targeting the backup pathway, PARP, that has become critically dependent on because the tumor suppressor is broken.
It's a very powerful strategy, and the source emphasizes that this concept provides a robust framework for performing large-scale genetic screens to systematically identify other synthetic lethal pairs, finding new drug targets that are lethal only in the context of specific mutations, either oncogenes or tumor suppressors, commonly found in cancer.
That seems like a huge area for future drug discovery. Okay, let's move beyond the DNA sequence itself and talk about epigenetic alterations. The source calls this the software of cancer, controlling how the hardware of the genes is used.
Right. Gene expression, whether a gene is turned on or off, isn't just determined by the underlying DNA sequence. It's also profoundly controlled by how that DNA is packaged with proteins into a structure called chromatin within the nucleus. Epigenetic changes are modifications to this chromatin structure or to the DNA itself that alter gene expression patterns. Importantly, these changes can be stable and passed down through cell divisions without changing the actual A, T, C, G sequence of the DNA code itself.
So it's like changing the instructions for reading the genome rather than changing the genome itself. What are the key mechanisms this source describes?
The main ones discussed are, first, DNA methylation. This involves adding a small chemical group, a methyl group, directly onto cytosine bases in the DNA sequence, particularly in regions called CpG dinucleotides, which are often clustered in gene promoter regions, the on-off switches for genes. Generally, high levels of methylation in a promoter region lead to gene silencing, turning the gene off.
Okay, methylation usually means off. What else?
Second is histone modification. DNA in the nucleus is wrapped around proteins called histones. These histones have tails that stick out, and various chemical groups can be added or removed from these tails, things like acetylation, methylation, phosphorylation. These modifications act like a code, influencing how tightly the DNA is wound around the histones.
And that affects whether genes can be read.
Exactly. For example, histone acetylation, carried out by enzymes called HATs, histone acetyltransferases, tends to loosen the chromatin structure, making the DNA more accessible and generally promoting gene transcription, turning genes on. Conversely, histone decetylation, done by HGACs, histone deacetylases, tends to compact the chromatin, making genes less accessible and leading to silencing.
So acetylation generally means on, decetylation means off.
As a general rule, yes. And these systems are interconnected. For instance, methylated DNA regions can recruit proteins that in turn recruit HDACs, leading to chromatin condensation and stable gene silencing.
And in cancer, these normal epigenetic patterns are completely disrupted, leading to chaos in gene expression.
Yes, absolutely. You get widespread, aberrant epigenetic changes that lead to the deregulation of gene transcription genes get inappropriately turned off when they should be on, and vice versa. A very common finding in cancer is hypermethylation, too much methylation, specifically in the promoter regions of tumor suppressor genes. This is a major mechanism used by cancer cells to silence these critical protective genes.
So instead of mutating the gene, they just switch it off epigenetically.
Exactly. The source lists several examples of key tumor suppressors frequently silenced by promoter hypermethylation in various cancers. P16, a CDK inhibitor, VHL, important in kidney cancer, BRCA1 in some breast cancers, STK11, P15, even genes involved in DNA repair like MLH1, which can cause microsatellite instability in sporadic colon cancer when silenced this way.
And it's not just hypermethylation, right? Hypomethylation also happens.
Yes. Widespread hypomethylation, too little methylation, is also frequent in cancer, often occurring in repetitive DNA regions, which can contribute to genomic instability. The overall pattern is just completely dysregulated compared to normal cells, and the changes in histone modifications are equally important, contributing to that altered landscape of gene expression.
The source notes that these epigenetic events seem to play a critical role very early in cancer development, maybe even initiating it, and are often found in premalignant lesions, and they can be influenced by environmental factors like smoking.
That's right. They're not just a late consequence. They can be fundamental drivers.
Yeah, but here's the crucial difference from genetic mutations like deletions. Epigenetic changes are, at least in principle, reversible.
This is a key point and offers a therapeutic opportunity. Unlike a deleted gene, which is gone forever, an epigenetically silenced gene is still physically there. It's just turned off. If you can reverse the epigenetic modification, say, remove the methylation or restore histone acetylation, you might be able to turn that gene, maybe a crucial tumor suppressor, back on.
And we actually have drugs that try to do this, targeting these epigenetic mechanisms.
Yes, we do. The source gives a great example from certain types of leukemia where chromosomal translocations create abnormal fusion proteins. For example, in acute promyelocytic leukemia, APL, the PML-RAF fusion protein abnormally recruits HDACs to gene promoters, silencing genes needed for myeloid differentiation and causing a block.
And how is that treated?
Remarkably, treatment with high doses of all-trans-retinoic acid, ATRA, a vitamin A derivative, can directly counteract this. ATRA causes the fusion protein to release the HDACs and instead recruit coactivators, including HATs, reversing the silencing and allowing the leukemia cells to differentiate into more normal neutrophils. It's a classic example of differentiation therapy driven by reversing an epigenetic block, although the source notes it can cause significant side effects, differentiation syndrome.
So that's targeting the consequence of the fusion protein. What about drugs that directly inhibit the epigenetic enzymes themselves, like HDACs?
Yes. HDAC inhibitors are a class of drugs. The source mentions vorinostat, romidepsin, and panobanostat as approved examples, mainly for certain lymphomas and multiple myeloma. These drugs block the enzymatic activity of histone deacetylases. By inhibiting HDACs, they generally lead to increased histone acetylation, which can help reawaken silenced tumor suppressor genes.
Do they only affect histones?
No, their effects are broader or pleiotropic, as the source puts it. Many non-histone proteins are also regulated by acetylation, so HDAC inhibitors can affect various cellular processes, promoting apoptosis, inhibiting the cell cycle, interfering with DNA repair, generating reactive oxygen species, disrupting protein-folding chaperones like HSP90. Their exact mechanisms are complex.
And what about targeting DNA methylation? Are there drugs for that?
Yes, DNA demethylating agents, also called hypomethylating agents. Drugs like 5-azacitidine and 5-aza2-deoxycytidine, decitabine, get incorporated into DNA during replication and act as inhibitors of the DNA methyltransferase enzymes that add the methyl groups. This leads to a passive loss of methylation over subsequent cell divisions, potentially re-expressing silenced genes. These are approved for treating myelodysplastic syndromes, MDS. The source notes limitations like poor solubility and bone marrow suppression.
And combining these approaches, demethylating agents plus HDAC inhibitors.
That's a rational strategy being explored, the idea being that both DNA methylation and histone deacetylation often cooperate to silence genes, so hitting both mechanisms might be synergistic in reversing that silencing.
The source also briefly mentions non-coding RNAs like microRNAs, miRNAs, and long non-coding RNAs, LNCRNAs, as being involved in this whole layer of gene regulation.
Yes, they are increasingly recognized as major players in controlling gene expression, often interacting with epigenetic machinery. Their expression patterns are frequently altered in cancer, and they represent another exciting, though challenging frontier for developing novel therapies.
Okay, let's pivot now to a totally different but equally fundamental aspect of cancer biology, metabolism, how cancer cells fuel themselves. They seem to have a unique way of managing energy.
Yes, altered metabolism is considered one of the core hallmarks of cancer. Cancer cells rewire their metabolic pathways to support not only their energy needs, but also the demands of rapid proliferation, constantly needing building blocks like lipids, nucleotides, and amino acids to make new cells, and also to survive in often harsh microenvironments and metastasize. And importantly, there's metabolic heterogeneity even within a single tumor.
And the most famous, most striking metabolic difference is the Warburg effect, also known as aerobic glycolysis. What's going on there?
This is a fascinating phenomenon first observed by Otto Warburg nearly a century ago. He noticed that many cancer cells, even when there's plenty of oxygen available, aerobic conditions, preferentially metabolize glucose through glycolysis, converting it primarily into lactic acid, lactate, rather than sending the pyruvate produced by glycolysis into the mitochondria for complete oxidation through the Krebs cycle and oxidative phosphorylation.
Which is weird because oxidative phosphorylation is way more efficient at generating ATP, the cell's energy currency, right? Why would they choose the less efficient pathway?
That's the paradox. While glycolysis yields much less ATP per molecule of glucose compared to full mitochondrial respiration, it does serve other purposes that are crucial for rapidly dividing cells. Firstly, it's fast. Secondly, and perhaps more importantly, running glucose through glycolysis allows the cell to shunt many of the intermediate molecules produced along the way off into biosynthetic pathways.
So it's not just about energy, it's about getting the building blocks.
Exactly. Those intermediates from glycolysis are essential precursors for synthesizing nucleotides, for DNA and RNA, lipids, for membranes, and non-essential amino acids, for proteins, all the stuff needed to build new cells rapidly. So the Warburg effect might represent a trade-off, sacrificing some ATP efficiency for faster production of biomass precursors.
And this high rate of glucose uptake in glycolysis is the basis for FDG-PT scanning and cancer detection, right?
Precisely. FDG is a radioactive glucose analog. Tumors that exhibit the Warburg effect gobble up much more glucose than surrounding normal tissues, so they light up brightly on a PT scan after FDG injection.
The source lists several key regulatory proteins involved in promoting this metabolic shift in cancer.
Yes, major players include oncogenes like CMYC and RAS, signaling pathways like AKT, the hypoxia-inducible factor HIF1, which gets activated in low oxygen but also by some oncogenes, and even tumor suppressors like P53 and PRB, whose loss can contribute to metabolic reprogramming. It's tightly linked to the other oncogenic signaling pathways.
And targeting this altered metabolism is also a therapeutic strategy.
It is, though perhaps still less developed than targeting signaling pathways directly. One approach mentioned is targeting components of the PI3K pathway, particularly mTOR. mTORC1 is a major regulator of cell growth, promoting protein synthesis and inhibiting autophagy, a cellular recycling process. Inhibiting mTORC1 with drugs like everolimus or temsirolimus can put a brake on cell growth and proliferation, partly through these metabolic effects. These drugs are approved for several cancers. The source also mentions the AMPK pathway, which acts as an energy sensor and normally inhibits mTORC1 when energy is low as another potential target.
Glucose isn't the only nutrient cancer cells are hungry for. The source mentions glutamine as well.
Yes, glutamine is another key nutrient that many cancer cells utilize heavily. It serves as a source of both nitrogen, for nucleotides and amino acids, and carbon, which can feed into the Krebs cycle for energy or biosynthesis. Like glucose, it's often metabolized in ways that prioritize building blocks. Cancer cells can be quite adept at scavenging necessary nutrients from their surrounding microenvironment.
And sometimes, mutations in metabolic enzymes themselves can be the actual drivers of cancer.
Yeah.
The source points specifically to mutations in IDH1 and IDH2.
Right, this was a major discovery. Mutations in the genes for isocitrate dehydrogenases 1 and 2 are found recurrently in certain types of brain tumors, gliomas, acute myeloid leukemia, AML, and bile duct cancers, cholangiocarcinomas. Normally, these enzymes convert isocitrate to alpha-ketoglutarate as part of the Krebs cycle, but the mutant IDH enzymes gain a new abnormal function.
What do they do instead?
Instead of producing alpha-ketoglutarate, IgG, the mutant IDH enzymes convert it into a different molecule called 2-hydroxyglutarate, 2-HG. 2-HG accumulates to very high levels in these cancer cells and acts as an ON metabolite, a metabolite that directly contributes to cancer development.
How does 2-HG cause trouble?
It turns out that alpha-ketoglutarate is a required cofactor for a large family of enzymes called dioxygenases. These include enzymes that are crucial for epigenetic regulation specifically, enzymes that remove methyl groups from histones, histone demethylases, and from DNA, TE enzymes involved in DNA demethylation. 2-HG structurally resembles AKG and competitively inhibits these AKG-dependent dioxygenases.
So the buildup of 2-HG messes with epigenetic regulation.
Exactly. It leads to widespread changes in histone methylation and DNA methylation patterns, particularly DNA hypermethylation. This altered epigenetic landscape disrupts normal cell differentiation processes, contributing significantly to the development of leukemia and gliomas. The discovery that a metabolic mutation could drive cancer primarily through epigenetic effects was really groundbreaking.
And we now have drugs that target these mutant enzymes.
Yes, specific inhibitors have been developed that block the activity of mutant IDH1, ivosidenib, and mutant IDH2. An azidenib note. Script previously said gilteritinib, which targets FLT3-AXL. These are approved for treating IDH mutant AML and are in trials for other IDH mutant cancers. They lower 2-HG levels and can promote differentiation.
Even metformin, the common diabetes drug, is being looked at in cancer trials because of its metabolic effects.
Yes, metformin primarily acts in the liver to inhibit glucose production, gluconeogenesis, and it also activates AMPK in cells, which, as we mentioned, inhibits mTORC1. Because of these effects on cellular energy metabolism and growth signaling, and some epidemiological data suggesting lower cancer risk or better outcomes in diabetics taking metformin, it's being actively tested in numerous cancer clinical trials. However, the source notes that its definitive role as an effective anti-cancer agent is not yet clearly established.
It seems the field of cancer metabolism is still uncovering many specific vulnerabilities that might be exploited.
Definitely. Understanding the precise metabolic dependencies of different cancers compared to normal tissues is crucial for developing effective and safe metabolism-targeted therapies.
Okay, let's move to what ultimately makes most solid tumors so lethal. Metastasis. The ability to spread.
This is absolutely central. The source highlights that metastasis, the spread of cancer cells from the primary tumor site to form secondary tumors in distant organs, is responsible for the vast majority, perhaps 90%, of deaths from solid tumors. Understanding how this happens and how to prevent or treat it is critical.
And it's described as an incredibly complex, multi-step process, not easy for a cancer cell to achieve.
Not at all. It's often called the metastatic cascade. It begins with a local invasion, breaking out from the primary tumor. The source describes 3 key features needed for this. First, the cancer cells have to change their adhesion properties to detach from neighbors and attach to the basement membrane, a thin layer separating tissues. Second, they need to locally degrade that basement membrane and the surrounding extracellular matrix, ECM, basically chewing a path through it, often using enzymes like matrix metalloproteinases, MMPs. Third, they need to actively migrate through that breached barrier into the adjacent tissue.
They also have to survive being detached, right? Normal epithelial cells usually die if they lose contact with their neighbors, and the ECM, a process called anoikis.
Exactly. That's another critical hurdle. Metastatic cells must acquire resistance to anoikis. They have to suppress this programmed cell death pathway that's normally triggered by detachment. The source also details a process thought to be important for invasion and migration called epithelial mesenchymal transition, or EMT.
Right, EMT. This is where epithelial cells, which are typically well-behaved, stuck together in sheets, and polarized, having a distinct top and bottom, undergo a transformation. They lose these epithelial characteristics.
Like losing cell adhesion molecules. Ecadherin is a key one.
And they gain properties more typical of mesenchymal cells, which are normally more migratory and invasive.
Yes, they become more spindle-shaped, change their internal scaffolding proteins, like switching from cytokeratin to vimentin, alter their expression of integrins, which mediate cell ECM adhesion, and become much more motile.
This EMT process happens normally during embryonic development and also temporarily during wound healing and fibrosis, right?
Yes, or it's tightly regulated and usually reversible. The difference in cancer is that the cells undergoing EMT often retain their unregulated proliferation capacity, and the transition might become more stable. This switch is driven by specific transcription factors like snail, slug, twist, ZEB, that are often activated by signals from the tumor microenvironment, such as TGF-beta or HDF acting through its receptor c-Met.
So EMT helps them break free and move. Is it required for all metastasis?
That's still an area of active research and some debate. The source acknowledges that while EMT is widely studied and implicated, evidence suggests that maybe not all metastasis requires a full EMT switch, and cells might metastasize through collective migration or other mechanisms. Its precise role and context dependency are still being elucidated.
Okay, so after invading locally, the cells have to get into the bloodstream or the lymphatic system to travel.
That step is called intravasation. Then they have to survive the journey in the circulation, which is a hostile environment, mechanical stress, lack of adhesion signals, and surveillance by the immune system. They travel to a distant organ, somehow get stuck in the small capillaries there, and then exit the vessel back into the tissue. That's extravasation.
And then the final, maybe hardest step, establishing a new tumor in that foreign environment.
Exactly. They have to survive and thrive in a completely new microenvironment, co-opting the local stroma and inducing angiogenesis to build a new blood supply. The source mentions that sometimes clusters of tumor cells traveling together, oligoclonal clusters, might be more efficient at seeding metastases than single cells, perhaps due to cooperative survival or signaling advantages.
And the source makes a key point. The real bottleneck, the rate-limiting step for successful metastasis, is often not getting there, but the ability of the disseminated tumor cells to survive and proliferate in the new metastatic site. This is heavily influenced by the specific interactions between the tumor cells and the host microenvironment in that target organ.
The seed and soil hypothesis revisited.
Yeah, it sounds incredibly difficult to develop drugs that specifically target such a complex multi-step process directly.
It has been very challenging because the critical molecular details driving each step, especially the later steps of colonization, are still not fully understood or are highly context dependent. However, the source notes that some existing therapies, particularly targeted agents or adjuvant therapies given after surgery, likely improve survival in part by inhibiting micrometastasis or aspects of the metastatic process, even if they weren't designed specifically as anti-metastasis drugs. Potential targets identified in preclinical studies include things like HER2, HIF factors, CMET, VEGFR, SRC kinase, FAK, MMPs, COX2.
And it's highlighted again that probably only a fraction of cells within the primary tumor actually possess the full metastatic potential. They need specific genetic or epigenetic changes to succeed.
Right. It might involve activating specific prometastatic genes, like those encoding chemokine receptors that help them home to certain organs, and/or inhibiting metastasis suppressor genes. Identifying these suppressor genes whose loss allows metastasis to occur, is really valuable. Modeling these processes in animals helps research enormously.
The example given of bone metastases really illustrates that crucial interaction between the metastatic seed and the specific soil of the host organ.
It's a perfect example. Bone is a very common site for metastases from cancers like breast, prostate, lung, kidney, and multiple myeloma, and it often causes significant pain and debilitating fractures. Normal bone is constantly being remodeled with a balance between osteoclasts, cells that break down bone, and osteoblasts, cells that build bone. This balance is tightly regulated, involving signaling systems like the rank-rank-lopage pathway.
And cancer cells mess this up.
They do. Cancer cells that metastasize to bone often secrete factors like PTHRP, various interleukins like IL-1, MIP-1A, that dramatically tip the balance towards bone destruction. They stimulate osteoclast activity and or inhibit osteoblast function.
Like in multiple myeloma, where the myeloma cells themselves directly activate osteoclasts and suppress osteoblasts, leading to those characteristic punched-out ilytic lesions you see on X-rays.
Exactly. And understanding this mechanism has led to effective therapies. Drugs that inhibit osteoclast activity, like bisphosphonates or denosumab, which is an antibody blocking AIR-ANKL, are now standard treatments to reduce skeletal complications, bone pain, and fractures in patients with bone metastases from various cancers. It's a clear success story of therapeutically targeting a specific host-tumor interaction that's essential for metastatic growth in that particular site.
Very cool. Okay, let's turn to another really important concept that helps explain why tumors are so resilient and difficult to eradicate completely. The idea of cancer stem cells and tumor plasticity.
Right, the cancer stem cell CSC hypothesis. This proposes that tumors aren't just a random collection of identical proliferating cells. Instead, similar to normal tissues, they might be organized hierarchically, with only a small subpopulation of cells within the tumor possessing the properties of stem cells specifically, the ability for long-term self-renewal, and the capacity to initiate and sustain tumor growth effectively.
So most cells in the tumor might be able to divide for a while, but only the CSCs can keep the tumor going long-term and potentially seed new tumors.
That's the core idea. The source uses examples from acute myeloid leukemia, AML, and chronic myeloid leukemia, CML, where experimental evidence suggested that perhaps less than 1% of the bulk tumor cells had this tumor-initiating capability in transplantation assays.
What are the characteristics of these hypothetical CSCs?
They're thought to share some properties with normal tissue stem cells. They're relatively undifferentiated. They have extensive, perhaps unlimited self-renewal capacity. They can divide to make more CSCs. And they also have the ability to differentiate, giving rise to the more specialized non-stem cell progeny that make up the bulk of the tumor mass.
But crucially, unlike normal stem cells, which are tightly regulated,
CSCs are unregulated. Their self-renewal and proliferation aren't controlled by the normal feedback mechanisms. The hypothesis also suggests that CSCs might arise either from mutations occurring in normal tissue stem cells or potentially from more differentiated cancer cells de-differentiating and reacquiring stem-like properties through processes like EMT. This highlights the concept of plasticity. Cells might be able to switch between stem-like and non-stem-like states.
And why is the CSC concept so important clinically, particularly for therapy?
A key implication, and a major reason the hypothesis gained traction, is the idea that these CSCs might be inherently more resistant to conventional cancer therapies like chemotherapy and radiation.
Why would they be more resistant?
Several potential reasons are proposed. CSCs are often thought to traverse the cell cycle more slowly, or be quiescent in G0, compared to the rapidly dividing bulk tumor cells that most traditional chemotherapies target. They might also express higher levels of anti-apoptotic proteins, like BCL2 family members, making them harder to kill, and/or overexpress drug efflux pumps, like proteins from the MDR-ABC transporter family that actively pump chemotherapy drugs out of the cell.
So the therapy might effectively kill the fast-growing bulk of the tumor, causing it to shrink.
But it preferentially spares the slow-cycling, intrinsically resistant CSC population.
And these surviving CSCs can then regenerate the entire tumor.
Exactly. They can self-renew and differentiate to repopulate the tumor, often leading to relapse, and the relapsed tumor might now be enriched for these resistant cells. This provides a compelling explanation for why many cancers recur even after showing a good initial response to treatment.
So the therapeutic goal then becomes specifically targeting and eliminating these CSCs.
That's the ultimate aim driven by this hypothesis. Identify unique surface markers or signaling pathways that distinguish CSCs from both normal stem cells, to avoid toxicity, and the bulk tumor cells, and then develop therapies that specifically eradicate this resilient population.
But the source also includes that important caveat about plasticity.
Yes. If non-stem cancer cells can readily gain stem cell properties, perhaps in response to therapy or signals from the microenvironment, then just targeting the cells that currently look like CSCs might not be enough. Achieving durable remission might require eliminating all cancer cells capable of regaining that stem-like tumor-initiating potential. The exact nature, origin, and frequency of functional CSCs across different tumor types and the degree of plasticity remain areas of intense research and some controversy.
This idea of plasticity, the ability of cancer cells to change their state, their phenotype seems absolutely central to the whole challenge of resistance.
It really is. Cancer cells, and perhaps especially CSCs, exhibit significant plasticity. They can alter their biology, their gene expression patterns, their metabolism, their sensitivity to drugs, in response to external pressures, whether it's chemotherapy, radiation, inflammation in the microenvironment, or attack by the immune system.
It's like they're constantly adapting, shape-shifting to survive whatever we throw at them.
That's a very good way to put it. This inherent plasticity, combined with the pre-existing heterogeneity within the tumor population that we talked about earlier, and the complex dynamic interactions with the tumor microenvironment, all of these contribute to the cancer's formidable capacity to adapt to therapeutic stress and ultimately develop resistance.
And the source lists just a daunting spectrum of mechanisms they use to achieve this resistance, both initially, intrinsic resistance, and after treatment starts, acquired resistance.
It's a long list. They can prevent the drug from getting into the cell in the first place, or actively pump it back out using those efflux pumps. They can increase enzymes that metabolize or inactivate the drug. They can alter the drug's target protein, mutate it so the drug doesn't bind, or just make way more of the target to overwhelm the drug. They can rewire their metabolism or signaling pathways to bypass the blocked pathway. They can alter DNA replication or repair processes to tolerate drug-induced damage. They can learn to evade the immune system more effectively. It's incredibly versatile.
And the harsh reality, underscored again in the source, is that for most metastatic cancers, with a few notable exceptions like germ cell tumors that are often curable with chemotherapy, resistance eventually develops to essentially any single therapy used over time.
It's almost inevitable with current approaches for many advanced cancers. Therefore, understanding the mechanisms of resistance and developing strategies to prevent or overcome it is arguably the single biggest challenge and thus a primary focus of contemporary cancer research.
Okay, let's bring the tumor microenvironment back into focus and delve into two more absolutely critical aspects of its interaction with the tumor. Angiogenesis, the process of building blood vessels, and immune evasion, how cancer hides from the body's defenses.
Right. We've touched on the microenvironment as a complex ecosystem. The source also specifically highlights the established link between obesity and increased risk for a number of common cancers. It lists breast, postmenopausal, colorectal, ovarian, endometrial, esophageal adenocarcinoma, gallbladder, thyroid, kidney, and others.
What are the proposed mechanisms driving that link between obesity and cancer risk?
It's likely multifactorial. Potential mechanisms include hormonal changes associated with obesity, like elevated levels of insulin, insulin-like growth factor 1, IGF-1, leptin, and altered sex hormones. Changes in the metabolism of adipose tissue itself, and crucially, the state of chronic low-grade inflammation often seen in obesity, mediated by inflammatory factors like IL-6 and TNF-alpha produced by fat cells and associated immune cells. The impact of obesity on cancer survival after diagnosis can be complex and sometimes paradoxical, depending on the cancer type and other factors. Now, angiogenesis. This is fundamental. Tumors simply cannot grow beyond a very small size, maybe a millimeter or two in diameter, without securing their own blood supply. They need oxygen, nutrients, and growth factors delivered, and waste products removed.
So they have to trick the host body into growing new blood vessels into the tumor mass. The source calls the point where this kicks off the angiogenic switch.
Yes, this is considered a crucial step in tumor progression. It's the point where the balance of factors in the tumor microenvironment tips in favor of vessel growth. Pro-angiogenic factors, signals that stimulate vessel formation, start to outweigh anti-angiogenic inhibitors. Stimuli that can flip this switch include hypoxemia, low oxygen in the growing tumor core, inflammation, or certain genetic lesions in the cancer cells like loss of p53 or activation of RAS.
And how do they actually build these new vessels? What's the mechanism?
They essentially subvert the body's normal processes of angiogenesis. They can co-opt existing nearby vessels, but more commonly they induce sprouting angiogenesis. Tumor cells, and also cells in the surrounding stroma, like fibroblasts and immune cells, start secreting pro-angiogenic factors. The most potent and well-studied of these are the vascular endothelial growth factors, or VEGFs, especially VEGFAs.
And VEGF signals to the endothelial cells lining existing nearby blood vessels.
Exactly. VEGF binds to VEGF receptors, VEGFRs, particularly VEGFR2, on the surface of endothelial cells. This triggers the endothelial cells to proliferate, migrate towards the VEGF source, the tumor, and organize themselves into new vessel sprouts that grow into the tumor mass. The source details other important signaling systems involved too, like angiopoietins binding to their Tie2 receptors and ephrins binding to EPH receptors, which help regulate vessel maturation, stabilization, and branching.
And those hypoxia-inducible factors, HIF1 and HIF2, that we mentioned in the context of metabolism, they play a big role here too, right?
A major role. Hypoxia, which is common in growing tumors, stabilizes HIF proteins. HIFs are transcription factors that then move to the nucleus and turn on the expression of a battery of genes needed to adapt to low oxygen, including, crucially, the gene for VEGF. So hypoxia directly drives VEGF production via HIF. Also, certain genetic defects, like mutations in the VHL tumor suppressor gene, which normally targets HIFs for degradation, can lead to chronic HIF activation and high VEGF levels even in normal oxygen, which is a key driver of the highly vascular kidney cancers associated with VHL syndrome.
So VEGF signaling through its receptors, especially VEGF2, is absolutely critical for tumor angiogenesis.
It is considered a master regulator. VEGFR2 activation directly promotes endothelial cell proliferation, migration, and survival. Other receptors, like VEGFR1, might play more nuanced roles, sometimes acting as a decoy or modulating the response. The angiopoietin system also fine-tunes things. Angiopoietin-1, binding to Tie2, tends to promote stable, mature, non-leaky vessels. Angiopoietin-2, ANG2, often upregulated in tumors, acts as an antagonist to ANG1, destabilizing vessels and making them more responsive to VEGF, which might be important in the early stages of sprouting.
Are there other factors involved besides VEGF and angiopoietins?
Yes, the tumor microenvironment is a soup of factors. Other pro-angiogenic molecules mentioned include basic fibroblast growth factor, BFGF, TGF-beta, TNF-alpha, IL-8, often produced by stromal or inflammatory cells recruited to the tumor. Integrin, those cell adhesion molecules, also play a role on the activated endothelial cells themselves. Specific integrins like AVA3, AVA5, and AVF1 are upregulated during angiogenesis and are important for endothelial cell adhesion, migration, and survival, sometimes interacting directly with VEGFR2.
But the blood vessels that the tumor builds aren't normal, healthy vessels, are they? The source describes them as chaotic.
That's a critical point with major implications. Tumor vasculature is typically highly abnormal. The vessels are often tortuous, dilated, chaotically organized, with excessive branching and shunting. They tend to be very leaky, with poorly formed junctions between endothelial cells and discontinuous basement membranes. They often lack proper coverage by supporting cells called pericytes.
And what are the consequences of this abnormal structure?
Several important ones. The chaotic structure leads to sluggish and uneven blood flow within the tumor, contributing to areas of severe hypoxemia and acidosis. This harsh environment can actually select for more aggressive, treatment-resistant cancer cell variants. The leakiness leads to high interstitial fluid pressure within the tumor, which paradoxically makes it harder for drugs delivered through the bloodstream to penetrate effectively into the tumor tissue. The lack of pericytes contributes to vessel instability. Some tumors even exhibit vascular mimicry, where aggressive tumor cells themselves form vessel-like channels.
And this abnormal vasculature can also help the tumor evade the immune system.
Yes. Changes in the expression of adhesion molecules on the tumor endothelial cells can hinder the trafficking of effector immune cells into the tumor. Plus, the hypoxic and acidic environment created by the poor vasculature can directly suppress the function of anti-tumor immune cells like T lymphocytes.
Given the reliance of tumors on angiogenesis, targeting this process has been a major therapeutic strategy.
Absolutely. The idea is that by blocking the formation of new blood vessels, you can essentially starve the tumor of oxygen and nutrients, inhibiting its growth and potentially causing regression. The source revisits agents developed based on this strategy, primarily those targeting the critical VEGF-VEGFR signaling pathway. Bevacizumab, Avastin, is a monoclonal antibody that binds directly to VEGFA, preventing it from activating its receptors. Ramucirumab is an antibody that blocks VEGFR2 itself. Zilvercept is a recombinant fusion protein that acts as a VEGF trap, soaking up VEGF. Then there are the small molecule tyrosine kinase inhibitors, SMIs or TKIs, we discussed earlier in targeted therapies. Many of them, like sunitinib, sorafenib, regorafenib, extinib, pazopanib, lenvatinib, cabozantinib, inhibit VEGFRs alongside other RTKs, so part of their action is anti-angiogenic. The source also mentions a newer inhibitor targeting HIF2 alpha directly, approved for VHL-associated cancers.
And combining these anti-angiogenic agents with other therapies, like immunotherapy, is being explored.
Yes, there's rationale for synergy. For example, by normalizing the tumor vasculature to some extent, anti-angiogenic agents might improve the delivery and function of immune cells or other drugs. However, these therapies aren't without side effects, often related to their effects on normal blood vessels. Common issues include hypertension, and rarer but serious risks include blood clots, bleeding, bowel perforation, and impaired wound healing.
Okay, finally, let's tackle the interaction between cancer and the immune system. Specifically, how cancer manages to evade which should be a powerful defense mechanism.
Right. The immune system plays a critical role in maintaining organismal integrity, constantly surveying the body for non-self or altered self, including pathogens and potentially cancerous cells. The fact that individuals who are immunosuppressed due to inherited conditions, organ transplants requiring immunosuppressive drugs, or HIV infection have a significantly higher risk of developing certain cancers is strong evidence for this immune surveillance function.
Both innate and adaptive immunity are involved.
Yes, both arms play roles. But the immune system needs to be tightly regulated. It has to be powerful enough to eliminate threats, but also restrained enough to avoid attacking the body's own healthy tissues, autoimmunity. Cancer cells become masters at exploiting these regulatory mechanisms, these natural brakes on the immune system, to hide or actively suppress an effective anti-tumor response.
So how do they do it? What are the main evasion mechanisms the source describes?
They employ a whole arsenal of tricks. A common one is downregulating MHC class I molecules on their surface. MHC molecules are needed to present tumor antigens, abnormal proteins, to cytotoxic T lymphocytes, CTLs, the main killers. Less MHC means less visibility to T cells.
They become harder to see.
Exactly. Another major mechanism is expressing inhibitory proteins on their surface, the so-called immune checkpoint ligands. The most famous are PD-L1, programmed death ligand 1, and the ligands for CTLA4. When these bind to the receptors, PD-1 on T cells, CTLA4 also on T cells, they deliver a strong inhibitory signal, essentially telling the T cell to shut down or become anergic, unresponsive.
It's like the cancer cell puts up a "don't attack me" or "I'm one of you" sign.
Precisely. They can also secrete immunosuppressive cytokines and factors into the tumor microenvironment, molecules like TGF-beta, IL-10, IL-4, IL-6, which directly dampen the activity of effector T cells and promote suppressive immune cell types.
And they actively recruit help.
Yes, they can recruit and promote the expansion of immunosuppressive immune cells within the tumor microenvironment. Key players here are regulatory T cells, TREGs, certain types of macrophages, often called M2 macrophages, and myeloid-derived suppressor cells, MDSCs. These cells actively suppress the function of cytotoxic T cells and other anti-tumor immune components. They can also induce tolerance in T cells or downregulate death receptors on their own surface to resist being killed.
And given tumor heterogeneity, these evasion tactics can evolve and change over time or differ between metastatic sites.
It's a continuous dynamic arms race, and those recruited inflammatory cells like macrophages and MDSCs can have dual roles, not only suppressing T cells, but sometimes even directly promoting tumor growth, proliferation, and angiogenesis themselves.
We've seen incredible breakthroughs with immunotherapy recently, but the responses vary hugely across different cancer types. Why is that?
That's a critical question. Some cancers, like melanoma, kidney cancer, Merkel cell carcinoma, certain lung cancers, bladder cancer, Hodgkin lymphoma, and cancers with high microsatellite instability, MSI high, often respond quite well to immune checkpoint inhibitors. Others, like pancreatic cancer or microsatellite stable, MSS colorectal cancer, tend to be much less responsive, often termed cold tumors immunologically. The reasons are complex and still being actively investigated.
What factors seem to predict whether a tumor is likely to respond to immunotherapy, especially checkpoint inhibitors?
Several factors are being studied and used, though none are perfect predictors yet. The source lists. Tumor mutational burden, TMB. The idea being that tumors with more mutations might generate more abnormal proteins, neoantigens, that the immune system can potentially recognize as foreign. The presence of these specific neoantigens. The expression level of the checkpoint protein PD-L1 on tumor cells or infiltrating immune cells. This is used clinically to guide therapy decisions in some cancers, like NSCLC, but it's not universally predictive. The density and type of pre-existing immune cells, particularly T cells, already infiltrating the tumor, TILs, and even the patient's own host genetics might play a role.
So we still need better biomarkers.
Absolutely. We desperately need more reliable biomarkers to predict who will benefit, who won't, and why, and also to guide the development of combination strategies to try and convert those non-responding cold tumors into hot responsive ones.
Okay, so how are we actually using immunotherapy now? The source discusses different approaches, generally activating the response, engineering T cells, and releasing the brakes.
Right. Older approaches involve trying to generally stimulate the immune system with molecules like interferons or interleukin-2, IL-2, which had some success but often significant toxicity. Modern approaches are more targeted. One cutting-edge strategy is CAR T cell therapy.
Chimeric antigen receptor T cells. That's where you engineer the patient's own T cells.
Exactly. You take T cells from the patient's blood, genetically modify them in the lab to express a synthetic receptor, the SARR, that's designed to specifically recognize a protein, an antigen, on the surface of the patient's cancer cells, like CD19 on B-cell leukemias and lymphomas. You then expand these engineered CRT cells to large numbers and infuse them back into the patient.
And these engineered cells then hunt down and kill the cancer cells expressing that target antigen.
That's the principle. It has produced remarkable durable remissions in patients with refractory B-cell ALL and large B-cell lymphoma, leading to approvals for these indications. It's truly personalized living therapy.
But it's not without risks. The source mentions significant potential toxicities.
Yes, CAR T therapy can cause severe side effects, including cytokine release syndrome, CRS, which is a massive systemic inflammatory response, potential organ toxicity, and neurotoxicity. These require specialized management and experienced centers. Resistance can also occur, sometimes because the tumor cells stop expressing the target antigen loss. Research is intensely focused on making CAR T therapy safer, preventing resistance, and extending its effectiveness to other blood cancers and, the big challenge, solid tumors. Allogeneic, off-the-shelf CAR-T approaches are also being developed.
The other major immunotherapy revolution has come from releasing the brakes, the immune checkpoint inhibitors.
This has arguably had the broadest impact across many cancer types. The strategy here is to use monoclonal antibodies to block those co-inhibitory checkpoint molecules that cancers exploit to shut down T-cell responses.
Blocking the don't attack me signals.
Exactly. The two main targets currently are CTLA-4 and PD-1/PD-L1. Anti-CTLA-4 antibodies, like ipilimumab, block CTLA-4, which primarily acts as a brake early in T-cell activation, mainly in lymph nodes. Anti-PD-1 antibodies, like nivolumab and pembrolizumab, and anti-PD-L1 antibodies, adalimumab, durvalumab, avelumab, block the interaction between PD-1 on T cells and its ligand PD-L1, often expressed on tumor cells or other immune cells in the microenvironment. This PD-1/PD-L1 axis acts more like a brake within peripheral tissues and the tumor itself, dampening ongoing T-cell responses.
And blocking these interactions basically takes the brakes off the T-cells, allowing them to recognize and attack the tumor cells more effectively.
That's the idea. Unleash the pre-existing or therapy-induced anti-tumor T-cell response.
And these drugs are now approved for a wide range of cancers.
Yes, they've dramatically changed the treatment landscape for melanoma, lung cancer specific types, kidney cancer, bladder cancer, head and neck cancer, Hodgkin lymphoma, MSI-high cancers, and others. Combinations are also increasingly used and approved, particularly combining anti-CTLA-4 with anti-PD-1, like ipilimumab plus nivolumab, has shown benefit in melanoma, kidney cancer, certain lung cancers, mesothelioma, and MSI-high colorectal cancer. Many trials are ongoing combining checkpoint inhibitors with each other, with chemotherapy, radiation, targeted therapies, and other novel agents.
And again, factors like high PD-L1 expression or high TMB can sometimes predict a better chance of response to these checkpoint inhibitors.
They are used as predictive markers, yes, although imperfectly, and the field is actively exploring other potential checkpoint targets, both inhibitory ones like LAG3, TIM3, TIG, and co-stimulatory ones like CD27, CD40, OX40, with modulating antibodies in clinical development, aiming to further fine-tune the immune response for better efficacy or broader applicability.
But unleashing the immune system like this is a delicate balancing act, isn't it? You risk autoimmune side effects.
It absolutely is. That's the major challenge with immunotherapy. By releasing the brakes designed to prevent autoimmunity, you run the risk of the activated immune system attacking the patient's own normal tissues. These immune-related adverse events, IRAEs, can affect almost any organ system. The source lists common examples like skin, rash, colon colitis, lungs, pneumonitis, liver, hepatitis, thyroid, pituitary gland, joints, etc. While often manageable, they can sometimes be severe or even life-threatening.
So a huge part of immunotherapy research is figuring out how to maximize the anti-tumor effect while minimizing or managing these autoimmune toxicities.
Precisely. Understanding who is at risk, how to treat IREs effectively, without completely shutting down the beneficial anti-tumor response, and finding that optimal therapeutic window is critical for making these powerful therapies safer and more effective for more patients.
It's clear that understanding the intricate dynamic dance between cancer cells and the host immune system is a rapidly advancing field with enormous potential.
And it holds immense promise for continued improvements in cancer treatment in the coming years.
Wow. We have covered an incredible amount of ground here, really diving deep into the biology from that single-cell origin, through all the broken cellular machinery, the signaling chaos, the metabolic rewiring, to the complex ecosystem of the tumor and its intricate interactions with the body's own systems like blood vessels and immunity.
It really highlights the staggering complexity, doesn't it? And the key takeaway is that all these biological aspects we've discussed, the cell cycle, apoptosis, signaling pathways, epigenetics, metabolism, the microenvironment, angiogenesis, immune evasion, stem cells, plasticity, metastasis, they aren't happening in isolation. They're all deeply interconnected, involving complex crosstalk in essentially all cancers. This inherent complexity and adaptability is precisely what makes treating cancer optimally so challenging.
But this deep understanding, this incredible wealth of information that we've explored, drawing from sources like Chapter 72, this is precisely what's driving the development of new, smarter approaches.
Absolutely. It's the foundation. By identifying the specific molecular vulnerabilities in an individual patient's tumor, understanding its unique pattern of mutations, its gene expression profile, its epigenetic state, its metabolic dependencies, its immune microenvironment, we are increasingly able to tailor therapies. Moving towards giving the right treatment to the right patient at the right time, aiming for maximal effect against the cancer and minimal toxicity to normal tissues. This personalized precision oncology approach is fundamental to modern cancer care and future progress.
And this deeper biological understanding also holds tremendous hope for something potentially even more impactful, much earlier detection.
That's a critical point. If we can reliably detect cancer-specific molecules like mutated genes in ctDNA, aberrant proteins, or specific metabolic signatures in blood or other body fluids at a very early stage, perhaps even before a tumor is clinically apparent or has had a chance to metastasize, that could have a truly dramatic impact on reducing cancer mortality. The biological understanding we've gained is enabling the development of the highly sensitive and specific technologies needed to make early detection a widespread reality.
So while the challenges, particularly resistance and metastasis, remain incredibly formidable and far from solved,
the detailed, intricate understanding of cancer biology that we've aimed to provide in this deep dive gives us the fundamental knowledge base. It's the blueprint from ongoing research for designing more rational therapies, developing smarter combinations, and ultimately for the continuing promise of overcoming cancer's profound complexity.
it really does feel like getting that glimpse behind the curtain, seeing the sheer ingenuity, albeit destructive ingenuity, of how this collection of rogue cells manages to subvert so many fundamental biological processes that are essential for normal life. It's both terrifying and awe-inspiring in a way.
And understanding that rogue ingenuity, understanding the enemy's playbook at that deep molecular level, is truly our most powerful weapon in figuring out how to defeat it.
This deep dive has definitely laid bare the sheer adaptability and resilience of these cancer cells. They constantly evolve, they co-opt our own body systems, they find ways around virtually every attack we launch. It really does feel like a relentless biological arms race.
It certainly can feel that way. And that maybe raises a crucial question, something for you, the listener, to really ponder as you integrate all this information. Given how cancer seems to dismantle, mimic, or corrupt so many fundamental aspects of normal cell and tissue behavior, deciding when to divide, when to die, how to use fuel, how to interact with neighbors, and the environment is the ultimate future of cancer treatment. Less about simply finding more ways to kill the rogue cells, and perhaps more about figuring out how to re-educate or reprogram the body's own systems, including the immune system and the microenvironment, to restore that lost control and reestablish normal tissue homeostasis.
That's a fascinating thought to really sit with. Thank you for joining us on this incredibly detailed deep dive into the masterful and often terrifying complexity of cancer cell biology. We hope this exploration has brought you much closer to understanding this disease with the depth and nuance of the professionals who dedicate their lives to fighting it.
Keep exploring, keep asking questions. The more we understand these fundamental mechanisms, the closer we get to truly conquering this challenge.